Okay, and last but certainly not least, we have Aiden Houck. Aiden is a rising senior majoring in biochemistry who spent the summer in Vanessa Wheeler's lab, and he comes to us from Boulder, Colorado. And I just want to interject, like when the students are done, we do have cake, so I hope that you'll stay and chat with them and congratulate them for a remarkable summer of science. And I'll come back up after that.
Sounds good. All right, so as Sue mentioned, my name's Aiden. I was working in Dr. Wheeler's lab this summer, and my project was looking at the instability of X-linked dystonia Parkinsonism and the hexanucleotide repeat in males and females. So a little bit about X-linked dystonia Parkinsonism, or XDP. It's a Mendelian neurodegenerative disorder endemic to the Panay Island of the Philippines. And this disease is caused by a hexanucleotide repeat of retrotransposition insertion into the intron 32 of the TAF1 gene, as you can see here. And this SVA region, as I mentioned, contains this hexanucleotide repeat of CCC-TCT. And the age of onset, based on a study done by Bragg et al. In 2017, was found that the age of disease onset is inversely correlated with the repeat length in individuals, meaning the longer the repeat, the earlier the disease age of onset tends to be. And so how do we measure how long a repeat is in an individual? And so the way we do this is through PCR amplification. And once we get a PCR product, we can submit it for ABI sequencing, and we get a modal allele here on this graph. This is the output. And so we can then say this modal allele here is at 53 repeats, and that's how many repeats this individual has. We can then, through the use of GeneMapper and online technology, measure the expansion peaks and the contraction peaks. And the expansion peaks are anything to the right of the modal allele, and contraction peaks are anything to the left of the modal allele. And so, based on a previous study done by the Wheeler lab and the Ozelius lab, published earlier this year, is they looked at the expansion index in different brain tissues as well as blood. And as you can see, the blood expansion index is significantly lower than that of the brain tissues. And so this data here caused the Wheeler lab to look at the differences in blood DNA expansion between males and females. And so as you can see, based on this data that has not been published yet, males tend to have higher expansion index in blood than females do. And so the hypothesis for my project this summer was that the expansion index is overall lower in females than it is in males, as it's dependent on an active chromatin state. And so we were going to try and test this through the use of a methylation sensitive enzyme in hopes to destroy an active X chromosome that contains this SVA region. And so in previous studies, the PCR was taking a product from an active X chromosome and an inactive X chromosome containing this SVA region. However, to prove this hypothesis or test this hypothesis, our goal was to destroy the active X containing this SVA region and only get PCR products from the inactive X. And so to accomplish this, there were a few hurdles we had to overcome. First, we had to identify a methylation sensitive endonuclease that would cut our DNA, and that was HHAI. But as you can see, the PCR primers that we were using in the past would only amplify the hexanucleotide repeat and not the cut site. So the next step was to then optimize a new PCR primer that would not only amplify the repeat, but also the cut site. After we had done that, we needed to test the enzyme activity to make sure it was actually cutting DNA and working before we tested our samples. And then once we did those three steps, we could move on to testing our hypothesis. And so this study had three controls. We had undigested genomic DNA, which is just male or female DNA that was put through the PCR to show that we could amplify the repeat. Next was mock digested, which was DNA mixed with enzymatic buffer as a control to make sure that we could still amplify the DNA after digestion conditions. And then both of those would give us PCR products. And then another control for our test was digested male DNA. And this wouldn't give a PCR product, as the SVA would be destroyed on the X chromosome. And then our test was our digestion. And an assumption for this project was that the cut site on the male chromosome was non-methylated, but on the females, it was methylated and on the inactive X. So first steps that I accomplished this summer was the optimization of a PCR. And so the first step in this was I tested the original primers, primer 1 and 2, on XDP containing genomic DNA from iPSCs. And I not only did this to ensure that we could amplify the repeat in this new cell line, but also that I knew how to run a PCR reaction, because that was going to be important. I then tested the new primer pair using the original PCR protocol, because might as well start when you already have something sort of working, start from there. And then I tested a temperature gradient, because that was close, but it wasn't quite right. And so what we got is we got 66 degrees to be pretty perfect for the annealing temperature. And I could confirm this through the use of comparing an ABI sequence, as I showed you earlier. And we have this modal allele of 53, and comparing that to an agarose gel. And so the sequence that we're amplifying with 53 repeats was expected to be roughly 760 base pair. And based on the agarose gel, we can see it's roughly there. The next step was to test the enzyme, make sure it was working so we could digest both male and female DNA. So my first step was doing an in silico digest, which produced thousands of DNA fragments when this genome was non-methylated. We also used lambda DNA as our digest control, which showed in these 2 wells that our enzyme is functioning and is digesting DNA. However, the HHAI digest of genomic DNA didn't result in an obvious smear like non-methylation sensitive enzymes I tested previously. And you can see those 2 wells here. And so at this point, it was unclear if it was the genome was methylated and it wasn't being digested, or if our resolution wasn't high enough on an agarose gel to see the digested products. So what I then did is took the genomic DNA that I had digested and ran it through my optimized PCR. And so my controls for this were using the undigested genomic DNA, as you can see in this well, which gives us our expected product. So we know our PCR is working. Next was our mock digested gDNA. And the expected outcome of this was something similar to our undigested genomic DNA, as the only difference is enzymatic buffer and it was incubated. However, it looks similar to our last control, which is our HHAI digested genomic DNA. And because this is male DNA, we wouldn't expect a PCR product, as the SVA region would be destroyed. And so you can see those 2 wells here, they look pretty similar. So based on this, we thought that it could be somehow the buffer conditions of the enzymatic reaction are affecting our PCR post-digestion. And so we tested this with a buffer spike in of genomic DNA mixed with buffer and ran our PCR. And as you can see that in this well, it's pretty clear that that's what was going on here. So currently what I'm working on is purifying digested genomic DNA after the digest, but before the PCR. And it's actually running right now. So in the next couple of days, we'll know if it worked or not. And then after that, once we get our expected PCR products, we can use GeneMapper to quantify our expansion index and compare male and female DNA. As well as one thing we want to look at on GeneMapper is the contraction peaks, because in Huntington's, their PCR affected, but it's unclear if that's also the case in XDP. No one's looked at that yet. And then doing methylation detection with PacBio sequencing would allow us to understand how our samples are methylated and how that might be affecting our PCR and enzymatic digestion. And then we can also attempt to come at our hypothesis from another direction by looking at an enzyme that would only cut methylated DNA. And that's a potential way we can approach this problem from the reverse. Oh, sorry, that got out of line. But I'd like to thank the Wheeler Lab and Alan for letting me work under him and ask all the questions, as well as Cami Gillis for being a sounding board for ideas, and Sue for putting this program together. So thank you.
Thank you all for coming and spending this time during your day to listen to these remarkable presentations. Congratulations to all of you. Every time I listen to these presentations, 10 weeks, They've been here 10 weeks. They sound like they've been here a year. So a remarkable amount could be accomplished in a relatively short period of time, thanks to all of you in the audience, all the people who worked with them in the lab and taught them so much. So, and I loved hearing all of you talk about your future work, because hopefully it will be your future work as you go on to graduate school or medical school or whatever you choose to do. And maybe, like so many people before, we'll see you around these halls again. If you find yourself looking for employment after you graduate from college, I'm sure that based on your presentations, we'd all be happy to have you in our labs in the future. So we have some cake. Please chat with the students. Join me once again in congratulating all of them.